This is a production of Cornell University.
Great. Thank you, Johannes, for the introduction. Thanks for everyone for being here. Yeah, so I will be going over the 3 chapters of my dissertation today, and all of them revolve around the concept of molecular diversity of soil organic matter and how decomposition affects it and how that affects its persistence. As you all know, soil organic matter is the fraction of soil that consists of plant or animal tissue in various stages of decomposition. Soil organic matter lends many physical, chemical, biological benefits to soil function. And another large function that soil organic matter provides is its ability to retain organic carbon. In fact, soils contain 3 times the amount of carbon as the atmosphere. And the balance between soil organic carbon and carbon stored in the atmosphere as carbon dioxide or methane is really dependent upon microbial respiration of that carbon. So it's actually a very old question of what retains, or what are the controls that are influencing the retention of carbon in soil. So it's such an old question. One of my favorite papers to go back to every once in a while and read is this paper from 1925 by Solomon Waxman, What is Soil Humus? And he asked this important question, which is really the driving question of my research as well, which is why does a part of organic matter decompose rapidly and a part only very slowly? He points out the economic importance of this question because at that time, really they're thinking of nutrients and nitrogen and the minerals that plants need to grow. For us, there's this added economic importance for climate change. So the current view of how we understand organic matter being cycled in soils is that there's this continuum. As organic matter is progressively decomposed, it becomes smaller and smaller. Along this continuum, it has the ability to chemically interact with minerals through absorption or desorption, and then also physically by being included or released from aggregates. So this process, along with some of the bigger environmental variables such as temperature and moisture, pH, soil microbial community, overall impacts the retention time of carbon in soils. More recently, though, we've been thinking about some other processes that affect organic carbon cycling, and broadly sort of the heterogeneity of soil itself as a control in soil carbon cycling. So there's spatial heterogeneity, so the co-location of organic matter, minerals, microbes. There's temporal variability, and then there's the molecular diversity, so variability in organic matter properties, which is, of course, what I am focusing on. So I define molecular diversity as the number and abundance of distinctly different compounds in soil organic matter. And being clear that I'm talking about diversity of molecules and not diversity of microbes right now. So the big question to my research is really, does molecular diversity affect decomposition of soil organic matter? I hypothesized that as diversity of molecules increases, we would have slower rates of decomposition because the energy return on investment by microorganisms to use each of these individual compounds would become unfavorable. They had to make new enzymes or upregulate new metabolic pathways to tackle each new compound that they come in contact with. So that's sort of the step 1 hypothesis, is that molecular diversity can slow decomposition. But that decomposition itself might increase molecular diversity as microbes add all these new compounds during their metabolism to the pool of organic matter. It might actually increase molecular diversity itself. So there's this feedback loop between decomposition processes and molecular diversity that I am also going to try to untangle. Some more foundation information. How am I measuring and quantifying molecular diversity? This is, there are studies that are looking at, they often call it hemodiversity, and those studies are more of a qualitative, and I really wanted to be quantitative, quantifying all of this. So to do that, I'm looking at the dissolved organic matter pool from soil organic matter, and this is where you do a water extraction, and you filter at 0.2 microns, and that's the pool of carbon. It is actually the most bioavailable and sort of usable by microorganisms. The next step is to actually identify those compounds. And to do that, I use untargeted tandem liquid chromatography mass spectrometry, so LCMS-MS. where I can then identify the different compounds based on mass to charge ratios and retention times. So you match those with known standard libraries. I use C18 and Hill columns for polar and nonpolar compound identification. This is just an example spectra where you have the mass charge on the x-axis, your intensity on the y-axis. It's from two different soil samples. But each of these different mass charge ratios could be considered a unique molecular compound, and then its peak height can be used to calculate the relative proportion of that compound in your sample. I use three big types of calculations to quantify molecular diversity, and these are really borrowed from ecologists trying to apply them to molecules. So the first one is just molecular richness, and this is just the number of compounds, and this is the most basic form of diversity quantification, which gives you really no information about the molecules, just the number. And it treats all rare and common compounds as one, so it's already equally weighted. The next one is an abundance-based molecular diversity equation, where then you take into account the proportion of the molecules across samples. So I use Hill numbers when the Q quotient equals 2, and this is synonymous with the Simpson diversity index, but it gives you effective number of compounds back instead of an arbitrary index. And this favors the common compounds. But again, this sort of lacks some information, so the next step is to add information about the molecular properties. So using a trait-based molecular diversity equation, I use Rao's quadratic entropy. And so the Hill numbers is a probability equation, this is an entropy equation, so it's different in that fact. And then also incorporate the distance matrix of a molecular property of my choosing. So it can be something like oxidation state or mass that gives us an idea of maybe how that molecule behaves in a soil system. So just as ecologists want to know the types of organisms there, so a gorilla and a lizard are very different in an ecosystem, and here saying a lipid and organic acid might also be equally different. So we need to know their properties when we're calculating diversity. I also use the terms alpha molecular diversity and beta molecular diversity, and I define alpha molecular diversity as a diversity within one sample, and beta molecular diversity as a diversity across samples, across treatments, across ecosystems. So when I first started my PhD, I did a lot of literature review on what did we know about molecular diversity in natural ecosystems. And really, the only field of scientists that were working on quantifying this were marine scientists or aquatic systems. Specifically, of course, in Dittmar's group has a lot of work around this. And I found this one paper where they did try to quantify diversity in the way that we did. And so this is a marine DOM incubation. And so they did molecular richness, molecular abundance, and then a trait diversity equation where they used the oxidation state of carbon. And really what they found is what we hypothesized, where there would be this increase in diversity initially, but then over time, it either plateaus or declines, and then there's this, decline in functional diversity in their system. So we wanted to know, so this is a marine system with continuous mixing and no minerals and no plant roots and it's all homogeneous. So what do we find in these same trends in a soil system? This is really where we got to my first chapter, which is really, do ecosystem properties or microbial decomposition affect molecular diversity? So this is really a step one proving ground of, does molecular diversity even vary across landscape in these very different ecosystems? And can we measure it too? And quantify it. So I used soil samples from 6 different ecosystems that we did as a larger collection effort with some postdocs in Johannes's lab. And I grouped these samples by vegetation class, so dominant vegetation class, and soil order. They're representative in 2 different ways. And you can see from these photos that they're very different from one another in their groupings. I also took samples from the litter layer down to the sea horizon so that I could use soil depth as a proxy for duration of decomposition. We know that organic matter found in the sea horizons is generally older than that found in the litter layer or A horizons. So this way we can use depth as a proxy for time. And I hypothesized that because microorganisms are the ones really doing all the action, they're the one, their transformations are going to have a greater control on organic matter molecular diversity than the plant matter inputs themselves, especially when you get down to the sea horizon. Jumping right into the results, here I'm showing hydrophilic and hydrophobic compounds identified using LCMS on the helicity columns. And for this project, I actually used negative and positive mode, and these are on water and methanol extracts. Sorry about that. And most notable thing about these plots, so this is molecular richness, and A, molecular diversity using abundance, whole numbers, and then the functional or trait molecular diversity equation in panel C, and depth is on the y-axis. You see that there's this decline in this alpha molecular diversity with soil depth. But the most noticeable decline really is from this litter layer, the litter samples, down to your first mineral A horizon. I also want to point out this very interesting trend for the functional diversity and with the hydrophobic compounds. I'm going to tuck that away for another chapter. But there's this initial increase and decrease with depth there. So if I separate these out by ecosystem, and I'm just showing molecular richness, you can see that generally there's always going to be this decrease in richness from the litter layer down to the A horizon. But there's variation once you get to the mineral horizons. And of course, there are a lot of different properties that soils have that could cause this. And for example, the coniferous forests and the grasslands, there's a large increase from the B horizon to the C horizon. And that holds true for the hydrophobic compounds as well. And so we thought, this must be some sort of environmental variable or soil property. And so I did a whole bunch of regressions with all the other properties that we had measured, such as soil texture, sand, silt, clay, hydroxylamine extractable, oxides, iron, aluminum, manganese, carbon concentrations, nitrogen concentrations, water holding capacity. We actually found no consistent and significant relationships between any of these properties in our molecular diversity indices. However, we did not measure any microbial information for this project. So I thought, okay, if there's this decrease in molecular diversity and richness with depth, then are all of those compounds that are accumulating in the sea horizon the same microbial products? They must be similar across ecosystems. So to try to answer that question, I did an upset plot analysis. And these plots look a little bit more complicated than they really are. So I'll walk you through the different panels. So there's the litter A and B sea horizon. So all the compounds are separated by horizon. Then I bin them into basically two different categories. One is, does that compound occur in all of the compounds denoted by these black dots, or is it only in one individual ecosystem? So the intersection size is the number of compounds, and then the ecosystems are also have the number of compounds that are within that individual ecosystem for that horizon or layer next to it. And they're all colored by compound classification. So for the litter layer, there are 471 compounds that were shared across all 6 ecosystems. And then by the time you get to, so the tundra sedges is our next one here, it has only 40 unique compounds in the litter layer for the tundra. That's how you read these plots. The most notable finding from these is that by the time you go from the litter layer to A horizon, there's already a 65% reduction in shared compounds. And then once you get to the C horizon, there's only 75%, or there's a 75% loss of shared compounds. So the litter layer really shares 4 times the number of compounds as the subsoil C horizons. And in this way, so we're losing richness, but we're actually increasing dissimilarity across ecosystems. This is further displayed by a non-metric conditional scaling plot, just coordinating the hydrophilic compounds. And you can see that the litter layer here, these dark green symbols cluster together really closely, whereas all the mineral samples kind of mishmash over one another. And in fact, the litter type or ecosystem type did explain 10% of the variance, but horizon explained 21% of that clustering. And to further drive the point home about the dissimilarity in the subsoils across ecosystems, so the NMBS does have a grade current dissimilarity matrix incorporated into it, and this is the average grade current dissimilarity index displayed by horizon to compare each ecosystem. And you can just see that there are more yellows and oranges occurring in the sea horizon, showing that there's just more of the similarity across ecosystems further into the subsets. So to summarize this chapter, and really this is the title tagline of this paper that came out last summer in PNAS, is that decomposition decreases molecular diversity and ecosystem similarity. In general, we found a decrease in alpha molecular diversity with depth, an increase in beta molecular diversity with depth, though. So the diversity across ecosystems increases. And in general, there are more shared compounds in the litter layer compared to the sea horizon, meaning there's probably more site-specific compounds that microbes are making in the subsoils compared to what's being inputted by the litter. So really, this is a study about molecular diversity across soil depth for time and time of millennia, long periods of time of decomposition. But really, I wanted to next now, like, okay, well, what about true time in time that I can measure in the lab? And would we find similar trends over shorter periods of decomposition time? How long did it take to get to these trends? So my next chapter is essentially that, is how does molecular diversity change over time? And can we identify the microbial products or the diversity of those products and how that contributes to those changes? And so to do this, I did a very simple, I'm calling it a long-term time series experiment, but really it's relatively short. It's only 220 days. PhD is only so long. And so I used one microbial community and one plant litter type and incubated that for 220 days and destructively harvested microcosms at 14 different time points. I then analyzed the DOM extract with the ATR, FTIR, and TOC, and then from there selected 7 time points and then analyzed using LCMS to quantify molecular diversity. So and then the next layer is then, how do I identify microbial products? To do this, we wanted to try to use an isotope label of O18 instead of a carbon substrate label, because there's some issues with using a carbon labeled carbon. We wanted to use O18. O18 is normally used by microbiologists to do stable isotope probing to identify which microbes are actually present in a community, because all microbes use water for the most part during their metabolism and respiration processes, and they generally take up the O18 and add it to their DNA, and then you do the stable isotope probing and figure out who's active in your community. We wanted to know, okay, well, can we then use it to know which compounds the microbes are producing or oxidizing, or maybe they're inputting some necromass that has this O18 in it. And we submitted a proposal to EMSL and got an EMSL exploratory research grant to explore this and try it. So I chose 2 different time points, day 6 and day 59, to then take a subset of the sample from the microcosms and added either natural abundance water or O18 for biotic and abiotic, just so it parse out which compounds are actually being produced with the O18. And then we did water methanol 4-form special extraction, but I'm going to only be going over the water extraction results today with Fourier transform ion cyclotron resonance mass spectrometry primarily. And again, I hypothesized that because the microbes are the ones, not only are they transforming the organic matter by cleaving all the different plant litter compounds and oxidizing, hydrolyzing, fermenting everything, they're making all these different metabolites, enzymes, and waste products in the organic matter. So the more active a microbial population is, the more diversity of compounds you will have. So these are the results from the LCMS long-term time series experiment. And what we can see is that these trends actually are very similar to microbial profile trends, where duration of decomposition is on the x-axis up until 220 days. Panel A is molecular richness, the number of compounds. We have then our abundance molecular diversity using Hill numbers. And you see these large declines in richness and diversity in the beginning of the decomposition. For this project, I use a multivariate molecular trait diversity equation where I use the nominal oxidation state of carbon, oxygen and carbon ratios, hydrogen and carbon ratios, number of nitrogen atoms and mass all in one equation to try to get this whole molecule picture. And what we found is that there's actually, instead of an initial decrease, there's an initial increase in molecular trait diversity. So diversity of molecular properties is increasing initially as microbes are decomposing the litter. I'm a big fan of Breaker's dissimilarity indices. So this is showing, again, the dissimilarity across time points. And the most notable thing, and you can see it here, is that you have these larger differences from day 2, 8 to 32. So bigger changes in the index, bigger changes in color. And by the time you get to day 60, this is really a plateau. There's not much change happening. And you can see that with the curves as well. So can't tell if that color is bad. The 018 results from the FTR-CMS. If you're familiar with FTR-CMS, you're familiar with these Van prevalent plots. These plots have oxygen to carbon ratios on the x-axis and hydrogen to carbon ratios on the y-axis. These are elemental, not mass ratios. I'm showing the top left plot is the 018 isotopically enriched compounds from day 7, and then on the right is are the non-isotopically enriched, and then the bottom two panels are from day 60. These plots, each point is an individual peak identified with mass spec, and then the coloration of that peak is the relative abundance across samples. Often with Van prevalence, you use some sort of grid to sort of try to understand the properties of that molecule. And here, there's some different zones. So this one is more, these compounds are more aliphatic. These ones would be more aromatic, mid-aromatic. You have low, mid, high oxygen regions. To the right, these are just calculated bar plots of the molecular richness of comparing the enriched to the non-enriched for day 7 and day 60. And then also Hill number diversity, and then our molecular trait diversity using a multivariate equation. Okay, complicated figure kind of explained. So the most important thing to take from this figure, well, not most important, but the most obvious one is that the non-enriched pool has many more compounds than the enriched pool. And I should note that the compounds that these enriched ones, they were incubated for 24 hours. So this is a snapshot of what microbes are producing within a 24-hour time frame at these two different time points. So non-enriched are more rich, more diverse. Not that surprising, they're just kind of the background molecules. But if you look at the molecular trait diversity, there's actually equal trait diversity when you're considering these peaks and these peaks at the early time point. So even though they differ in number, the trait diversity is just as large. However, by day 60, there's a decline in trait diversity. You can see that there's some loss of these aliphatics. Well, the aliphatic region seems to be lost, but the overall trait diversity between day 7 and day 60 does not differ. And if we go back to our time series plot, these two time points are essentially the same as these on the longer scale. And so it's kind of like we're picking up on maybe these shoulders of trait diversity for day 7 and day 60, and we're kind of missed that initial peak for the overall background. Or also that maybe there's a lag where the microorganism community starts making fewer and fewer diverse compounds, and then it takes a little while for that to be shown in your overall curve. Okay. Also, looking at what compounds are being produced and consumed over these two time points, day 7 and day 60. This is just a heat map of the different compounds identified with LCMS for day 7 and day 60. This is adjacent to the 018 labeling experiment. The brighter colors mean they're more abundant, a higher relative abundance. And so what we're seeing here is that the sugars and sugar alcohols at day 7 are occurring in higher abundances. And then at day 60, there are more of these organic acids, meaning that perhaps microbes are, consuming these carbohydrates from the fresh plant litter inputs, and then producing a whole bunch of organic acids during their metabolism. And this is just sort of an overall snapshot of what I think is happening for this chapter, where as you introduce microbes to plant litter, there's going to be this initial decrease in molecular richness during rapid microbial growth. They're going to be consuming all these different simple sugars, but they're producing a wider range of compounds. So this could be they're producing amino sugars, organic acids, like peptides, et cetera. And that's where you would get this increase in molecular trait diversity. So you're getting this increase in the different types of compounds that you're finding in the pool of organic matter. But with no new added plant litter or substrates, microbial growth is going to ultimately slow down. And then they're going to have to start recycling some of their own microbial products. And that's where you would get sort of this decline in molecular trait diversity. Everything comes back down to baseline, and microbes then start producing more and more molecular similar compounds. So at this point, maybe just all microbial products. So this was a study for one plant litter type and one microbial community. And my next question is really, does this hold true for different plant litter types and different microbial communities and microbial diversities? So there is an established relationship between microbial and molecular diversity, and we've seen it using this paper in 2022 that is sort of established across different biomes, and the relationship strength is varied across biomes. But really, I wanted to know, okay, is this established relationship true with different plant litter types and for different microbial communities? So this is my third chapter. Type and microbial community diversity and molecular diversity of UM. So to do this, I used 5 different plant litter types. These are from similar samples used in my first chapter. And then from the same location where I took the plant litter type, I took the A horizons, the neural A horizons, did a soil wash to generate a microbial community inoculum. So this paper is really plant litter type and microbial community are synonymous. You cannot sort of disentangle that relationship. But so from that soil wash, then I have this high microbial diversity community, which is 10 to 0, my stock community. And then we did a serial dilution of those communities to 10 to the minus 10 to get this low microbial diversity community. And I say we because my undergraduate, Caleb Levitt, and I worked on this for almost an entire semester, just getting these communities together. It was amazing. Okay, so then the plant litter that is complementary to that microbial inoculum was inoculated with either the high or the low microbial community. Incubated for 150 days, and then at the end of that incubation, the DOM was analyzed for molecular diversity using LCMS again, and the microbial community was analyzed using 16S rRNA sequencing using the Illumina MiSeq platform. And I hypothesized that, again, it's more about the microbial community structure than it is about where they came from. But again, those two are difficult to untangle. Thankfully, these dilution treatments had an effect on the endpoint microbial community. So these are the results from the end of 150 days, not the starting point of the inoculum, but the end of the incubation. For microbial richness, microbial diversity, and microbial phylogenetic diversity. And I am talking about microbial diversity now and molecular diversity, so I'll try to help you keep them separate. For microbial, more of this trait diversity, I'm using a microbial phylogenetic diversity index using Browse Project Entropy again. But for this, I'm using the genome size, the number of base pairs, the average number of base pairs per sample. And this is trying to get, again, an idea of that function of the microbial community in the soils, not just who's there and how many of them are there, but what are they doing? So there's about a 40% loss in microbial richness for 4 of the ecosystems, and then arid shrubs is just, a Death Valley sample. It's very unique and just kind of a bugaboo. but it was not affected in terms of richness, but it did, a portion of those individuals was altered. And then we have differences in phylogenetic diversity as well. Just ornating the communities and by litter type and microbial dilution treatment. It's the PCA plot showing how they cluster and just showing that the litter type more significantly explains the variance of the clustering than the dilution treatment, which did have an effect, but you can notice that the dilution treatment actually brought the microbial communities closer together in terms of composition. And if you're looking at the soil order, the composition, the relative composition, you see generally this loss in Ophidobacterialis and Burkholderia. I say loss, but it's compositional. And there's generally more Sphingomanolas and Gethmonolas. I'm so sorry for my pronunciation. It's a new language for me. Starting a suite of linear regressions. So here I'm plotting my microbial diversity indices on the x-axis against my molecular diversity indices on the y-axis. So I have microbial richness and molecular richness have a significant relationship here to R squared of 10. The microbial diversity using Hill numbers is significantly related to the molecular diversity with Hill numbers. And then actually genome size, rather than the diversity equation with genome size, was very strongly related to the molecular trait diversity. So there's something about the microbial community's function that is related to trait rather than just the number of microbes, which is, I think, more often what people think about is microbial richness is related to diversity in general. So to account for the dilution treatment and the litter type, community type, I did a multi-level correlation analysis where it takes into account dilution and litter type as random effects into the model. And that just kind of proves the point that genome size is the most strongest predictor of molecular trait diversity. And even though this interesting trend where richness is related to richness and diversity to diversity still holds true, but weaker. I noticed something else different when I was looking at these regressions is that the non-diluted communities actually appear to have stronger relationships with each other than with the molecular diversity indices than the diluted communities. If you look at molecular richness here on the x-axis, it actually is related to all of the indices, but this relationship with molecular richness is greatly improved. Same for microbial diversity and molecular diversity, and then similarly for genome size, the R squared here is 0.66 for molecular trait diversity. So there's something happening here that I find very interesting, and I don't necessarily have the answer yet about the disruption caused by my artificial dilution to try to get this bigger range of diversity has disrupted the microbial community functioning somehow. And it could be they're taking out the, members of the community that are involved in co-metabolism, or thus like kind of the whole chain disrupts. But these are just final musings. But in general, genome size is still the best predictor of molecular trait diversity for any diversity. Okay. So I'm gonna wrap up a little bit on this big question of does DOM molecular diversity influence organic matter mineralization rates? That's where I started. And I have not explained this experiment yet, but at the end of the incubations for my second and my third chapter, I saved the DOM from those endpoints. When I sent it off for LCMS analysis, I held them back. I then took that DOM and added it into a new microcosm, carbon normalized across each microcosm, which had clean sand, and then added one microbial community to that microcosm, and then measured respiration over the next 10 days. So this is cumulative respiration on the y-axis, and then either molecular richness, my molecular abundance diversity, or trait diversity on the X on the top panels, and then molecular properties on the bottom panels. So the oxidation state of carbon, molecular weight, or degree of saturation, which is a hydrogen carbon ratio. And I lumped together the two projects because they're essentially the experiments were done in the same manner. And overall, what I found is some weak correlations that I don't really believe in for molecular richness and diversity, which are just really being pulled by a handful of samples. But there actually does seem to be some pretty good relationships between molecular properties and the respiration rates of that DOM. Whereas with the oxidation state of carbon, you can imagine that as these values become more positive, that compound is more oxidized, therefore more readily available to be used, in comparison to some of these more negative values that are a more reduced compound, which have higher energy yield but are more energy costly to use. And same principle for molecular weight, where the bigger the compound is, potentially the more difficult it is to be degraded. But these are average values for an entire pool of carbon in a sample. So yeah, interesting. So coming back to this feedback loop of decomposition and molecular diversity. And really, what have I learned about decomposition and molecular diversity over the last 6 years? Overall, decomposition in the short term increases molecular trait diversity. Once there's a fresh plant litter supply, rapid growth, and all these new compounds are being made. But over time, without any new plant litter additions, you ultimately get this decrease in the number of different compounds and trait diversity over time if there's really no new food sources spurring microbial activity. Molecular diversity is influenced by microbial diversity and community functioning, potentially. And it's influenced by litter type and the fact that, that could cause faster, slower growth perhaps, but litter type inputs don't actually determine the endpoint molecular diversity of DOM. So just because, litter A has a higher starting point molecular diversity than litter B, it doesn't mean that litter A, after 200 days of decomposition, is still going to have a higher diversity than litter B. It has more to do with the microbial population. And ultimately, that microbial molecular diversity may influence organic carbon persistence, but there are truly some big hurdles to overcome in terms of instrument capabilities and some experimental barriers to really getting at this question, because we're talking about small molecules that we're trying to identify all of them in one pool and quantify them. It's very challenging. All right. I just want to say thank you, and I want to thank, of course, my committee members and all of my collaborators or people that have helped me along the way, let me use their lab, let me use their equipment, given me advice. And then my funding opportunities that I've had while I've been here. And I just also want to shout out for the Schmidt-Novak Small Grant Program. That has been, I wouldn't have been able to do this research really without that in the beginning. And the Cornell Center Graduate Research Grants also largely funded this research. And so with that, I'll take any questions.
Your conclusion about the type of development the molecular diversity, that and not only that, because you measure the organic composition actually processed by the microbial, so that's why it's called that molecular diversity of the organic matter is determined by microbial population other than the type.
Right. Wait, sorry, can you repeat the question? Yeah.
My question is that that's because you only measure the molecular diversity of dissolved organic carbon, which is usually processed by microbial. It's kind of the product of microbial process.
Right. DOM essentially is, especially off of a fresh plant litter, anything that you measure is already going to be like a DOM, microbial product, because the abiotic decomposition plant litter is probably quite small, and what we're measuring is really all microbial products from DOM. Is that what you're getting at?
Yeah, probably what you measure is they are all microbial process product.
Mm-hmm.
Yeah, and then probably they are also kind of the other
Yeah,
You can't.
I don't.
point to the phone.
Yeah, everything that we measure, so this is one of the challenges with the LCMS, is everything that we measure has to be filtered at 0.2 microns. And then the instrument itself actually only measures between like 200 and 1,000 Daltons. So everything below that is what we're measuring. You would have to sort of do some sort of bulk analysis where then you can quantify molecular diversity on that. And we did try some diversity calculations on NextApp spectra or FTIR spectra, but it's very difficult. Yeah.
Questions. I had a question at the very beginning in the depth profiles. We sort of eyeballed that there is in the hydrophobic compounds there's an increase in functional diversity. Can you speculate why we see it there and not for the hydrophilic compounds? Does that have anything to do with the analysis or production? Is that a true ecological phenomenon or mixing or movement? Or
one thought about that is that for the hydrophilic, they're hydrophilic, so they're more polar and they can move through the soil profile probably better. But the hydrophobic ones are really more in place. They're sticky to minerals and all that stuff. Potentially why we're seeing that trend sort of last longer with the hydrophobic compounds and not in the hydrophilic ones that are more transient.
Okay.
Just go back a couple of slides.
Yes.
I'm just getting a little bit nervous. I'm a little clueless here, but when the title says it's the big question and the two experiments really occupy different parts of the graph and drive the relationship.
Yeah. So I did these experiments separately for each project, but when I analyze them separately, they have different trends a little bit, which made me nervous about making any declarative statements about those trends when they differ by project. So I wanted to lump them together and see. These are normalized by the amount of carbon that was in the microcosmos too. So yeah, they're different.
Can you really draw a line through like experiment A and B together when they're just one's at the bottom left and the other one's at the right? I don't know. That just makes me wonder. Okay. Anyway, that's just
yeah. This is more just sort of musings on the big question and my best attempt to answer it with the data that I have. Yeah.
There's so much that's fascinating here. I'm sorry about that. Listening to this, but you mentioned something about the dilution disrupting the whole functional communities or something. Like how the microbial diversity might be working together to get the job done.
Yeah, this is something I hadn't considered when I was doing these zero dilutions, that I could just be completely disrupting the function of that community through that process, even though it's already quite a disruptive process to get to my stock community. The next step appeared to really disrupt any relationships. So my thought process was, we're thinking about this after lab group meeting recently of, well, if a whole community, every individual has a role in producing a certain metabolite or product that then this organism uses, and then it's like that chain reaction. They're all like working together. Then you take out some of those species, then you don't have the next person to eat that, and then that could be what's disrupting that trend in terms of diversity of molecules and biodiversity.
So far, this is where I couldn't quite see through, but are you saying that you feel your observations support that idea that the community does have this sort of pass along collectivity?
It's possible. I have to look into the data and look at more of the metabolic pathways that are present. I can do that and see if I'm losing certain pathways that would then maybe be like the next step. But it's my best assumption.
You had 5 different ecosystems, or some nice number at the beginning in chapter 1. Were they replicated? Like, did you really see that each one of them is somehow coherent microbial phenomenology, or is there sort of all sorts of variation within as much as across the system?
For the which chapter?
For the equal one ecosystems.
Yeah. Okay, were the 3 replicates sort of representative?
Yeah, they sort of go together. What was the coherence?
Yeah, there were definitely some variations within those triplicates because there's good difference between some of them. But we tried to lump them in the best way possible where the soil order was the same and the dominant vegetation type and general climate. So some of them were better, they had lower variances in general, but I think the forest here, which one had a lot more variation.
It's the molecular satellite diversification you're talking about. Is that more related to that? Or is there kind of like a way to distinguish that?
In these samples, there's not a way to distinguish between depth and time because we didn't measure like C14 on any of them, and that's also really difficult to do on DOM anyway. But so it's really just using depth as a proxy for time, and assuming that what's deeper is older.
Is there a way to kind of estimate that based off like the
Potentially, you could use. I mean, in general, you think about the samples that are in the northeast are going to be a lot younger than those in the southeast. So there's going to be an age discrepancy already that I know maybe you could use some accounts for, but I have not.
Last question. That's not the, oh yeah, Christina, go for it.
This was great. Thank you. I enjoyed it. Ripping off, I guess, what Rebecca was saying, do you think that the dilution step maybe lowered genome size, which would account for the lower trait diversity?
Doesn't appear that the dilution actually lowered the average genome size for the most part. You have some oddballs out here which are right in this relationship, but they're kind of in the same range, maybe slightly lower. I'm not sure if it's genome size. Yeah.
What do best dilutions do to the community and how can we do?
Right.
And what does it mean that these are correlated with diversity of molecules?
Yeah.
But it's obviously there's a difference. Somebody has a good idea.
Yeah.
Rachel would appreciate
Would love to talk more about this, but this is more of just like the interesting finding where I thought I was creating like this perfect, diversity gradient of microbes. Consider the ramifications in the community. Yeah.
Yeah. Great. Thank you very much, Ray.
This has been a production of Cornell University, on the web at cornell.edu.